So without further ado, I'd like to introduce our last presenter. It's a pleasure of mine to introduce a good friend of mine, Dr. Matt Nonnenmann. Dr. Nonnenmann is an associate professor up at the University of Iowa. You can see he's got his Iowa Hawkeye colors on. He is an expert in field aerosol exposure assessment and exposure-controlled methods, not only in agriculture, health, and safety, but he has branched off into other industrial sectors, including healthcare. So without further ado, Dr. Nonnenmann, you have the floor. Matt, you're muted. There you go.
Everyone hear me okay? Great. So let's see, I have the share content. So the quick question. So I think, is the presentation uploaded here and you guys advance for me, or do I just share my screen?
We can do it either way. Which do you prefer?
I'm happy to share my screen. That's
okay. Yeah, so that's on that bottom panel, just like we practiced last time. Just hit share.
Okay.
And if you already have your PowerPoint pulled up, you can either share content or share screen. There we go.
Okay, sounds good. I'm going to click the presentation mode here, and we'll see what happens.
Okay.
Can everyone still see my presentation?
Yeah, I'm saying yes. Now I see the first slide, so you can go ahead.
Great. Thanks, everyone, for the opportunity to present today. So again, my name is Matt Nonnenmann, and I'm an associate professor at the University of Iowa. And as Dr. DeFreight mentioned, traditionally my work has focused more on the agricultural side, looking at bioaerosols, dust control bioaerosols in the last 10, 12 years. And then and then I've been doing some work in healthcare. So I'm going to kind of give you a couple of examples of some work we've done in healthcare and also in agriculture, measuring some bioaerosols, and then talk about SARS-CoV-2 or some just that may work for SARS-CoV-2, because that seems to be the question, right? Is it aerosolized, or is it isn't, or is it not, and when and where? What's the concentration? Those sorts of questions. So we'll talk a little bit about that. And feel free to stop me during the presentation, if you have a question.
So, Dr. Nonnenmann, just a short note. I see a gray box on my side, so you may need to put this in a slide reader mode instead of presenter mode.
Sure.
Sorry.
No, that's okay.
And then all questions will be typed in, so they can just enter those questions into the Q&A box as you are presenting.
Okay.
And then we'll get to those at the end of your presentation. Dr. DeFreight will help moderate those.
Great. Okay, so I guess you'll see. Well, the good news is you'll kind of get to see what's coming, right?
Yeah. Why don't you click on the bottom of your? It looks like a little book on the icons on the bottom next to notes and comments.
Yep.
Reading view. There you go. Click on that and let's see what happens. Yep, that looks great. Go ahead.
Perfect. Thanks so much. So one of the big questions or concerns, right, is of course, related to how far an aerosol can transmit from a virus. And what we do know is, there's some evidence about Ebola. Often the recommendation is, 1 meter or 3 feet for Ebola, because in theory, it's more of, quote, the droplet transmission, risk for transmission. And obviously, if you think about something like measles, which is highly infectious, one of the most infectious viruses on the planet, the aerosol transmission could potentially happen even further. And so the question is, for SARS-CoV-2, how far is far enough? And so that, it's a fascinating question. And obviously, this is related to size and many other factors, size of the aerosol and many other factors, many other environmental factors. Dr. Mena pointed out some really important ones, like temperature, relative humidity, and the presence of sunlight or UV sources, And of course, if you're in an enclosed environment, there's concentration that's emitted and also the rate of ventilation, and all those variables come into play. And of course, whether someone is wearing a cloth face covering, which of course, as many folks know, provides a barrier, an impaction barrier for the larger stuff. So lots of discussion around this issue. And if we have time, I'll go into what's been happening locally in my community. So, okay, a little bit of introduction again. So bioaerosol behavior principles apply to bioaerosols. So, if the larger particles have those quote ballistic type properties where, they come out and they drop over time. And so same thing happens in bioaerosol. So it's, often considered a mixed suspension of liquid or solid particles. On the agricultural side, we'll see lots of solid particles, manure, bedding, that sort of thing. And the healthcare side, often we're thinking of like secretions or, sneezing, coughing, those sorts of things. So, and here's just kind of a scale to help us understand bioaerosol size. So on the x-axis here, we've got a couple of different things. We have, as you move to the right on the x-axis, you have settling time based on size. And you can see also here we have, size of particles in microns also on the x-axis. And so kind of the inhalable size is considered about 50% of 100 microns and lower. And so inhalable, that's a human exposure term describing a distribution of particle size that can be inhaled. And then when you get down to kind of respirable and smaller particles like coarse and fine particles as defined by the EPA, obviously the smaller you get, the settling time increases. And of course, that's what we're concerned about, the small stuff that can stay airborne for a while. An important point is that often viruses, they're not aerosolized in an individual level. So you're often, that's what we think, right? It's like chunks of dried saliva, or it may not be completely dried. So anyway, there's lots of discussion and lots of variables. But I think, the recommendation for 6 feet right now is a good one. So when we think about sampling, I mentioned this earlier, there's a, couple things to think about. So size selective sampling, so an inhalable sampler. You collect an aerosol that represents what people would inhale. And then, of course, when you're targeting bioaerosols, often the approach is dictated by what sort of data you want to get out of your analysis. And so if you're looking to sample virus and be able to assess the infectivity of that virus, that approach may be a little different than if you're looking for detection or looking for quantification. So often, just very similar to, say, targeting bacteria, we'll use like a nutrient agar, to sample. We don't really do this for bioaerosol samplings. We'll use a similar sampler, an impaction-based sampler, using, say, like a cell culture media or something that can go into the infectivity assay. So that's one approach I've seen. Also, other approaches using liquid-based sampling. So you have, this is kind of a classic bioaerosol sampler, the SKC bioaerosol sampler, and it has like a, a swirling action in here as you collect the sample. This sampler to the right of that, another type of sampler, and I've got a little bit more information about that, but it's kind of a more advanced, higher volume type sampler, also liquid-based. And then lastly, something that industrial hygienists are very familiar with, and that's filter-based sampling. So you can use filter-based sampling for virus, to look for airborne virus. And then lastly, the analysis to quantitate. I mentioned culture-based. So this can be basic microscopic examination, counting of colonies after growth for bacteria, molecular methods using DNA or RNA amplification, like polymerase chain reaction, and then also infectivity assays like a plaque assay for virus. Okay, I just have a few slides here showing you some work that we're doing. So we have one of the questions as an industrial hygienist is when you sample, what filter material should you use? And there's some data out there guiding us. The PTFE filter material has been the focus for virus for many years. But, you know, I'd argue that are some potential alternatives to that. And so, like, here's a filter that we had made on campus, and this is a spun polystyrene filter, and we spiked it, and we were looking for a surrogate virus, a murine norovirus, in this filter material. And I'm just going to kind of zoom in. You can see the scale down at the bottom, but it's a pretty good visual to see, you know, how these viruses are captured. I'm zooming in here on the filter material, and as we zoom in even further. You know, here, this is, you know, there's a lot of, often there's space on these spun filters. And, you know, here they have like properties to collect particles. And here we've zoomed in. This is 500 nanometers, really small. And we believe this is our murine norovirus. So there's lots of questions about this. But me thinking as an industrial hygienist, you know, and wanting to use techniques that can apply in many places and many times, so we're not so specialized to do the sampling. I think these filter data are pretty important to guide future work. So this is some work that we've done. So I wanted to show you some good visuals on that. Okay, so I kind of mentioned a little bit about the analysis of viral aerosol samples. So again, the plaque assay is an infectivity assay, and that's kind of almost, I would call it the gold standard of infectivity assays for virus. And so basically you have to be able to grow a cell culture in your lab that the virus can grow on. And then you collect your sample and you apply it to your cell culture, and you count areas where they call it plaques, but basically that's where the cells die because of viral infection. And so you can quantify that. The challenge is that it's, you know, the precision on the quantification is limited by error, and so when you compare the ability to quantitate to like a PCR reaction, you can get much more specific on a PCR reaction, looking at the genetic, the number of copies in a sample. The challenge is you don't know if those copies are active virus or inactive virus. So a great way to run these samples is to, if you can, collect the sample, do PCR on them, quantify them, say, wow, okay, we've got a lot of virus on this sample, go back and do a plaque assay. That's a great way to do it if you can, but could be freeze-thaw cycles in there. As Dr. Mena mentioned earlier, how does that impact the virus? You know, there's some challenges to figure that out. The flip side is to collect the sample and apply it to a plaque assay, all of them. But the challenge is these plaque assays are a lot of work. So on the molecular side, we have, you know, like qPCR, quantitative PCR, that gives us the number of viruses in a sample. And then also a droplet digital PCR, which is much more sensitive than qPCR, and you can use that. And the great thing about that is that you don't need a calibration curve, which is kind of nerdy talking about molecular stuff, but that helps sometimes. And then lastly, you can do sequencing. And so this is important to identify, you know, the whole genome sequence of that virus to see if it's, you know, related to other viruses that are being collected in the community or in the same building, and from, you know, a prevention standpoint. And also, it's always great to get an idea of what the sequence is, so you can have that. And you can also do this with bacteria and other biologicals. So we've done a little bit of that work. Okay, so talking about what we've done. So Dr. Mena mentioned some toilet aerosol work. We've done some of this in the hospital. And this was a really interesting study. And kudos go to my student, Sam Knowlton. And basically, we measured aerosol coming off of toilets from patient waste during flushing. And how we did it was we designed a cart that had pumps in it, high flow pumps. And we hooked up our impactor samplers here at a distance, and we would put the edge of the toilet right up to the front here. And my student would sit outside a room and wait for the patient to use the restroom, and then wheel the cart in there, get everything set up, and do the sampling, and then of course remove the cart. And it's very challenging in a healthcare environment. People don't feel well. They don't like noise. They like to use the restroom when they need it. And so we design things to be very quiet and be highly mobile, so we can just get in and get out very quickly, so we weren't interrupting patient care. A little bit on the results. It's a fairly simple analysis. We did just a total bioaerosol concentration measurement, which is colony forming units per cubic meter, across a couple of conditions. We had a background where there was no waste in the toilet. We didn't flush it. Then we had also a category where we flushed the toilet with no waste in it and did a measurement. And then we also had fecal waste in the toilet with flushing, and we did measurements there. and we identified that when there's waste in the toilet and you're flushing, we're measuring more bioaerosol coming off the toilet. Not a revolutionary observation, but we demonstrated that our methods do work. And subsequent to this, I won't click on the link, but we also completed a study targeting C. diff. So our goal was to design this cart, do some sampling, and then target specific patients who had very specific diagnoses, so that we could see if we could find that aerosol, which may be contributing to soiling of the environment and, of course, transmission. And, our initial goal was looking at C. diff, and we did find it in this particular paper. We found a few other things too. So if you want, you can go look at that. Okay, so another thinking, that was bacteria. So now thinking about virus. So we used, again, when I think about virus and measuring virus, I like to go where I know the virus is. And so like in this case, there were an influenza outbreaks on swine farms, and we called veterinarians who go in there and treat the pigs. And we're like, hey, can we hang samplers on you when you go into the farm to try and measure this virus in breathing zones? And basically, we use this research-based bioaerosol sampler. The air comes in and it swirls inside based on size, moves over to this smaller tube, and then finally onto some filter material. And so you end up with 3 samples from one measurement event, and these are separated based on size. And so some results, as you can see over here, we have the farm number. We have days elapsed from initial evaluation, like how long it had been since the pigs have been diagnosed. And these are these 3 stages of that sampling device. The filter, the smaller tube, and then finally the large tube, the 15 cc tube. And this sampler has a lot of advantages because you're not trying to extract off of, for all the stages, you're not trying to extract off a filter. So that's only in the final stage. And then you can combine the results and look at total copies. So here we detected the highest concentration, the earliest from the diagnosis, so a day or 0 days or 2 days. We were able to, and we compared it to another filter-based sampler, and we were able to calculate concentration. And again, this is RNA copies, not infection. Okay, so some interesting definitions. I've talked about viability, and basically this is when you sample, you collect an organism. If it's viable, it's capable of living or developed or germinating under favorable conditions. That's what that means. Culturability is often described, and we think about it from a bacteria standpoint. So the ability of something being able to be in culture. And this is challenging with environmental samples because it's not just one organism you're sampling many, and often there's virtually always, there's some sort of culture bias. So you see what on your growth, on your agar, you see what stayed alive during the sampling process, what stayed viable, and then you also see what is growing, and usually see what grows the fastest because it'll outcompete the organisms in the sample. And so often we'll go to our DNA or RNA-based analyses because it's a little bit easier on us to target specific organisms. But we don't know this information, which is important. Okay, so I'm just going to go through a few slides here describing. So I went to Europe with my family, visited London, and this was, like when everything just really happened, right? Everything was happening. It was very eerie there because, places were very empty, and we're like, are we doing something we shouldn't be doing? It was kind of nerve-wracking. But we got home. There was one line in Heathrow. That was it. It was quiet. It was empty. And then when we got home, my wife had a fever 5 days later. And so I'm like, oh my gosh, we need to sample. So I did a bunch of sampling around here. You can see I used those NIOSH bioaerosol samplers. She's binging on Netflix there, which everyone does when they're not feeling well. I also used this Coriolis sampler. You can see the spinning media there in the sampler. And here I just have a brief video kind of showing you what it looks like. Hopefully that's translating well. You can see the air comes in here. My arrow's off a little. The air comes in here and it swirls. And then you can analyze that liquid however you want using PCR or culture-based. So the results of this were we didn't find anything. We found no virus. And at that point, we couldn't get tested. Testing was not available. And so we were uncertain. Did she have it or not? And so now what we're doing in healthcare is we are sampling for virus aerosols during patient care, and we're using multiple approaches to try and find the virus. And we have had some success, but it's been very challenging because, of course, when you're using kind of a sampler that, in theory should work really well, the sampling duration is relatively short. And during that time, are there aerosolization events that occur in healthcare? Those are kind of the real questions. When do these events happen? What are people doing when these events happen? And is it patient based? Is it a cough? Is it a sneeze? Is it blowing their nose? What is it? Or is there activity, so like suctioning or specific healthcare activity that's occurred during those events? And so this work is ongoing, and hopefully we'll get some results out soon. Okay, so really important. This is the challenge, right, with bioaerosols, is that, there's very little information on what concentration is considered bad or we should not exceed. And this is continuously the challenge, whether it's bacteria, whether it's virus. And so, of course, the question is, what is the infectious dose of these viruses? And when are we controlling it enough? Or do we need to not detect virus at all? And if we detect virus using molecular methods, does that mean there's a hazard? And so there's lots of questions around these data, which we'll learn more about. But Dr. Minham mentioned earlier something about culturing the virus, and you can do that in a BSL-3 lab. I don't have one of those. We have one on campus. We're busy right now, but it is a challenge. And ultimately, I think, this culture or not to culture, that's kind of the question, right? So when you're collecting these environmental samples, it'd be great if we could do both all the time. And I would like to, but, it's resource intensive and it's access intensive. So, and then of course, the infectious dose for SARS-CoV-2, we don't know what that is. For examples like norovirus, it's very low, 18 viral particles. It's like, wow, that's so low. And so what is it for SARS-CoV-2? We don't know. Okay, with that, I will stop. I know we have time for questions, and thank you for listening.
All right. Thank you, Matt. Great presentation. Very, very interesting. Do we have any questions among our participants for Dr. Nonnenmann, or really any other presenter that we can follow up on? As you might expect, Dr. Nonnenmann is where he is. You probably heard Iowa is kind of a hotbed right now with new cases, and so he's dealing with some dynamics there in his community. All right, I don't see any questions.